Let's start again. I would like to introduce Professor Pier Mario Biava, who is an occupational physician, a researcher. He doesn't want me to say that, but I'd like to say it anyway. He's a scientist, and he works in Multimedica Group in Milan, and he will talk about his extraordinary research about stem cell differentiation stage factors, which is something unique, and he's been working on this for a long time, and I think it is unique research. So listen to him because he puts his heart in his research. Thank you. Thank you to Dr. Di Modugno for this invitation. Today, I'm going to talk about the relationship between stem cells and neurodegenerative diseases and cancer, and how the factors which we can take from zebrafish, a tropical fish, taken as a study model for stem cell differentiation stage factors, can function in the reprogramming of cells, while in order to prevent a lot of diseases such as the neurodegenerative diseases, and also in the age processing prevention, so a lot of functions. Discoveries have been progressive, and at the beginning my interest was on environmental carcinogenic factors. After taking my degree, I started studying how environmental carcinogenic work in inducing a tumor, and with a series of thanks to a series of experiments, I understood how these carcinogenic factors could work during pregnancy. Literature told us that if these factors enter into play when the different or during organogenesis, tumor arises in children. So what happens during the organogenesis? From a one-type cell, that is the oocyte, different cell stages bring to multipotent, pluripotent, and oligopotent cells until we have differentiated cells. When these differentiation stages happen, there are some substances which can correct the damage induced by environmental carcinogenic factors in stem cells, meaning that they can repair the damage which causes malignant tumors. I formulated two hypotheses. First of all, do these factors really work like this? Can they actually prevent the alteration of stem cells? Second hypothesis, are tumoral cells actually similar to alterated stem cells, so that the alterations in genome in some stem cells can be repaired? These were the two starting points of my research, and I needed to demonstrate the their truth. As I said, we take this zebrafish embryo, which is the most studied model for the cell differentiation of stem cells worldwide, because we know exactly when these processes and these regulating processes happen. So we are certain that in those moments there are differentiation elements. So we took this embryo at five different stages in the stem cell differentiation. You will be able to understand why we can detect these processes after my explanation about the gene expression. So, taking the embryo in those moment, in those exact moments, and being able to stabilize the process, we were able to determine the fact that experiments could be repeated. And we wanted to grant to be able to take the same factors and the working of these factors in the same way. So we started what it was all about, so what we were talking about. 98% of low-weight molecular, it is represented by these factors, and we analyzed the molecular weight of these proteins, which have low weight, because it is inferior to 40 kilodalton, whereas the other 50% has a weight which is comprised between 50 and 97 kilodalton. They are small proteins which can be absorbed under our tongue, and we utilized these factors by making the tongue absorb them. Going in depth with these studies with mass spectrometry tests, we detected the single proteins which belong to that method. You can see different series of these proteins, a complex network of proteins with a low molecular weight. They are very important proteins for the mitochondrial metabolism, that is to say for cell breathing, and they are really important in cell signaling. They are shock-thermic proteins. There are three types of shock-thermic proteins, which are really important for our immune system, and so they are really important for the whole cell metabolism. These proteins are the same one we can find in the human body, which means that when we apply a treatment, we don't use different substances, and 98% of the proteins in zebrafish are similar to the proteins found in a human body. So we treated the different human tumors, and we treated them at first in vitro. We treated different types of cancers, such as glioblastoma, which is a really aggressive tumor, brain tumor. You need to multiply the coordinates for 10,000 by 10,000, and then we followed the different growth curves. As you can see, the proteins of the first phase of differentiation, when a totipotent stem cell differentiate in three pluripotent, that is to say ectoderm, endoderm, and mesoderm, and the factors which are responsible for this differentiation block 80% of these tumors. Here we have skin cancer, which is blocked in 40% of the cases, and it is even more aggressive than glioblastoma. Then here we have mammary cancer, breast cancer, which in this first stage is not aggressive because the growth and control curve grows more slowly, and this curve is inhibited by 40%, but with factors which are taken during the more advanced stages. These kind of cancers respond in a better way during these last phases. We have here acute lymphoblastic leukemia, which is inhibited by a factor of 100%. And then we have the kidney adenocarcinoma, which is inhibited for 35%, but only at 48 hours. We have an explanation for this, but if you don't understand it, it's not a problem, because it's quite difficult to explain in detail all these molecular mechanisms. But I'll explain them to you anyway. These are the same tumors which were treated with intermediate and advanced stages of stem cell differentiation. So you can see that curves are basically repeated. There's a glioblastoma, skin cancer, breast cancer, and you can see that a differentiated, more benign tumor answers responds better to these last phases. In fact, at 24 hours, we can see that the process slows and that tumors recedes. The tumor recedes. And as a part of the cells starts a progress of apoptosis. You can see all the tumors, and this is the kidney adenocarcinoma, which repeats the same curves. But why does the process slowen? The starting point was that these factors are able to correct the genes which are responsible for the synthesization of the different proteins, which means that they correct and regulate the gene expression. We had to demonstrate it. This is the quantitative method to evaluate this oncosuppressor, P53. You can see these lights. These are the cells of glioblastoma before the treatment with our factors. And in this slide, you can see the red blotches, which are the proteins transcribed by P53 genes, and it is so called because its molecular weight corresponds to 53 kilodaltons. This protein, which is the concentration of this protein, increases after the treatment in treated cells. This means that our factors managed to regulate the P53 gene, which is an oncosuppressor gene. This gene was activated and transcribed, and it translated a protein, that is to say, the protein corresponding to P53. This protein managed to block the cell cycle of tumor cells, and then it tries to repair the damage which originates the malignancy of the tumor, and the damage is repaired if it is not too severe, which means that tumors are reversible. They can actually be cured. Otherwise, if damage is too extensive, genes which codify for cell death are activated and cells effectively die. We've managed to see these two phenomena because we were able to study them. We saw some proteins which increased by 100%, and we also saw an increase in the apoptosis. We also see an increase in stem cell differentiation and in cell programmed death. This is a more complex inhibiting mechanism of tumoral growth, which happens in kidney adenocarcinoma. It was not possible to regulate the P53 gene, and the duplication was blocked, so we tried to understand why. We tried to study another important molecule for the control of a sickle cell, which is the protein of retinoblastoma, which is one of those rare tumors which can be present when you're born. It is a tumor which can be inherited, and this is due to the alteration of a protein which is changed, and this is an important factor for the control of the cell cycle. This protein blocks the cell cycle when it is not phosphorylated. If it is phosphorylated, it detaches from 2F gene, which is transcribed, and it makes the cell cycle start with. So it depends on whether it is phosphorylated or non-phosphorylated. And it is useless for me to explain this slide, but we managed to show that with our factors we are able to regulate proteins which are already present and translated, the amino acid sequence, and we managed to change the form of this protein from phosphorylated to non-phosphorylated. And this non-phosphorylated protein manages to block the cycle. You can see that a lot of molecules change their behaviors if you change their conformation. We blocked the cycle by taking away the by dephosphorylating the molecule. If we have time, I'll explain to you this model, which is a cancer model, which allows you to understand everything about tumoral diseases. In a few words, the concept is the following one. There are 5 differentiating stages, and in every stage, from a progenitor cell, such as the oocyte, from totipotent oocytes, 3 different pluripotent cells derive: ectoderm, endoderm, and mesoderms. Because of these 5 stages, if we produce a model in order to understand how many differentiated cells are there, and we repeat these models for 5 times, the total number of differentiated cells is given by multiplying 3 for itself for 5 times, and we have 5 different types of cells, which are the somatic cells of our body. If we add to them germinative cells, that is to say, cells which originate oocyte and sperm, which are 9 different types of cell, gonium, primary, secondary oocyte, spermatogonial cell, spermatocyte, spermatide, and sperm. We have 252 different types of cells. And do you know how many cells we have? 252 cells, differentiated cells. So a simple algorithm can organize our life. 5 stages which originate these 252 different types of cells. So how many tumoral diseases are there? If tumors are indifferentiated stem cells present before the last differentiating jump, the number of tumor is given by 3 plus 3, and with this calculation, this algorithm, with the addition of all the spermiogenesis cells, gives us 130 different diseases, tumor diseases, and we can know the malignancy of these 130 diseases. There will be 3 really malignant and aggressive tumors constituted by pluripotent stem cells, then 9 by stems constituted by multipotent alterated stem cells, then 27 by oligopotent stem cells, and then we have 81 well-differentiated cells, which are the ones which respond better to this therapy, because, as I said before, these cells don't respond to chemotherapy and radiotherapy, and that's why we talk about chemoresistance. All these types of cells, as you understood, can be reprogrammed by way of these factors which regulate the gene expression, because they constitute the code which originates life and which switches on and off different kinds of genes, so that in the end the genome of these different 252 different cells, that is to say the DNA responsible for the protein synthesis, is always the same. The codefined DNA is the same in every single cell. So what changed in a brain cell if compared to a liver cell, which presents a different form? There are two different phenotypes, but the DNA which codifies for them is the same. So what changed? Genes changed because some different genes were switched on and off. So in the brain cells, in the brain, where different type of genes were active if compared to liver. So cell differentiation is a specific and selective gene regulation which makes it so that some of these genes are switched on and some are switched off, and the differences between the cells in our body are called epigenetic, because there's a code which regulates our life. It's called epigenetic code, which is above DNA, regulates all the genes of DNA, which are then responsible for the protein synthesization. And as I told you before, we took these proteins and we managed to produce some products which we then studied from a clinical point of view. This first study on 179 cases of primary liver tumor and hepatocarcinoma, and these patients couldn't respond to traditional therapies. So these patients were abandoned and left to their own means. And at the hospital where I was working, I had a very famous oncologist, and he was studying different phases of the hepatocarcinoma, and he managed to change the natural history of this disease, because usually from the moment of diagnosis to the moment of death, 3 or 4 years were usually passed, whereas he managed to increase the survival time by 2 years. This cancer, however, is multifocal, which means that new nodules are produced, but he couldn't understand what he could do. So he came to me and told me, what can we do? Let's see if your treatment works. He was really skeptical at the beginning, but I was lucky. And one of the first cases he saw, and which motivated him on this clinical investigation, which began in 2001 and finished in 2004, which means after 40 months, and which means that we had made a lot of in vitro experiments, and we could propose a model for the in vivo control. So this cancer is at an advanced stage. In the seventh part, you can see white nodules which represent the cancer. Then we have a portal thrombosis, which implies a bad prognosis of 8-10 months. And you can see that with this portal vein thrombosis, blood cannot pass. And after the treatment, the thrombosis disappeared and everything disappeared. And this really impressed him, because this was one of the first patients he saw. We made checks every 3 months, and our follow-ups were really controlled, were really detailed. My colleague saw this patient, and this patient, after 3 months, was treated. He had an alpha-fetoprotein, which is a marker of this tumor, which at the beginning corresponded to 300,000, and at the end it was only 3. And when he underwent an angiotac, everything had disappeared. if compared to the interventions which were made before, there was a new kind of result. A new tissue substituted the tumor, and this tissue is healthy. It is not different from the healthy part of the liver. And this effect was called evanescent effect, which means that this therapy is a differentiation therapy, meaning that it is able to reprogram tumor cell, and it makes them go back to normality in a physiological way. This is another case of multiple tumors which completely regresses. We had a regression of 20% and non-progression which was of 16%. And you can see that putting them together, we have a variation with a survival rate which is extremely significant. After these experiments, I realized that I had been extremely lucky in studying the epigenetic code at the beginning of life, because I could understand that only then, when life origins, the whole code is present from the beginning to the end, which means that all factors are present which are able to regulate all the genes of all the cells of our entire body. So I decided to further my understanding in order to understand what could happen by using the different parts of the epigenetic code, which is the so-called life code. When the differentiation cells process ends, it is any longer possible to study the whole epigenetic code, because in every organ, there is that part of the code which is necessary to control the physiology of that organ, but not all factors are present. Only in the organogenesis period, the one I had to study, all these factors are present.
So I can study the whole code or regulating life. And so what happens now? What does this code do? I wanted to study it, and so this code could be useful for any pathology of, those are different functions of stem cells. So here you find different stem cells. I already talked about pluripotent, multipotent, and so on. There is a specific division of those cells, which is different between the initial phase and the final phase, but that's not important. Here you can see from an hematopoietic tissue, only blood cells differentiate. No, many other cells differentiate. Connective tissues, bone tissues, cartilage tissues. From just one multipotent cell, many different cells develop. So now I was lucky to find and to work together with other professors in Roma. On the tissue renewal, I was lucky to cooperate with colleagues of mine from University of Bologna, who were recognized by the health ministry as a national point of reference, coordinating 23 universities. So we carried out those research under 26 Italian universities. So we worked all together. This study, so we carried out this study using the mesenchymal stem cells, which are adult stem cells that can be taken from those tissues you can see, adipose bone, tendon, and so on. We took them from the adipose tissue with a method which does not involve chemicals to pick them up, to pick those adult stem cells. So we took them from the adipose tissue and we treated them in different ways. So all those are the various employments of those factors in all those diseases. So bone marrow cancer, osteoarthritis, heart attack, cirrhosis, and so on. Many different diseases in which those stem cells can be employed. But of course, medicine in those diseases does not, follows the most complicated ways. So medicine wants to solve those diseases with a transplant of stem cells. That's a possible way, but it's very complicated. It requires complicated surgery intervention. This is the one in Parkinson's disease, where stem cells has to inject it in the substantia nigra. And this intervention is the one for so for the heart attack surgery. A stent is inserted and then stem cells are introduced, but the results are not that positive. So this is the traditional approach. But studying the positive evidence due to the transplanted stem cells, we found out that those positive effects are not due to the transplanted stem cells, but to the produced factors. And what are those factors? They are the same factors that we use and we get from the zebrafish embryo and make the different stem cells differentiate. So we thought at this point that instead of treating those pathologies by transplanting stem cells, we could just use the factors that we have in our body. So what would happen? So this is what happens. So this is an employment in beauty, like anti-aging moisturizers. But let's talk about neurodegeneration now. We carried out a study using the hippocampus cells, which are the first one degenerating in Alzheimer's disease. We led them to degeneration with two different concentrations of NMDA. So, and we could, thanks to instruments, we could detect the death of those cells using a fluorescence substance. Those in red are dead cells, while the black ones are the cells which are still alive. So those cells have been treated, and that's the result. And we got how life works from this exam. If we so using the whole network of the 5 stages, the results, of course, are positive, but not that significant. We would block the neurodegeneration by 20-25%. So we thought that to block the neurodegeneration, we first have to employ the whole network, so simulating the process giving rise to life using the whole 5 differentiating phases. Doing so, we learned that this way we prevent more than 70% of neurodegeneration. So the scientific paradigm used by medicine nowadays is wrong. So detecting the single molecules and not the whole totality is a limited approach because it does not get to great results. To reprogram a cell and regenerate a tissue and stop multiplication, we have to provide them with multiplication programs that are the same used by life. together with many pieces of information, all the pieces of information required, because only this way can the cells understand what they have to do. If we lack those pieces of information, the results are weak, are poor. So the opposite of what medicine is being doing now. So that's a complete change. Can you hear this noise? This is a sound of a cell at 22 Celsius degrees. As the temperature increases, the sounds are pure, but they increase in acuteness. So this is even more acute and pure. The last one. This is a dying cell. From a pure sound, the sound becomes noisy. So the new paradigm is based on the concept of information. At the center of everything, at the base, is a specific piece of information that directs cells to their destiny, to their fate. And this information has to be complete, total, and it can be transmitted through molecules. But cells can be directed to their fate even just using electromagnetic fields at low frequency and intensity. So we're not using molecules, but fields, electromagnetic fields. And we saw that cells interplay throughout molecules, but as well throughout sounds and throughout electromagnetic waves. Everything matters in life. Everything is information. At the basis of life, there is an informed energy which behaves in a complex way, not simple as medicine used to think. So the last point is the anti-aging. And we have no time to focus on the last thing I wanted to talk about, which is the new model of mind and body connection, but we have no time to talk about it. So here we're just focusing on the anti-aging process, and application. Now you get that there's a part of the epigenetic code that blocks aging, which is the very beginning part of the differentiating process. Because if I analyze this step before the differentiation, all those factors that can keep the stem cell gene on. So we took that part and we treated the mesenchymal stem cells of the adipose tissue in adults, and we activated the genes blocking the telomeres cut, which is the part which is being cut at in any multiplication, so that after a certain number of multiplication, the telomerase is completely cut. In this case, we keep the genes, the stem genes on, and those stem genes are the same genes that Yamanaka had introduced thanks to a retrovirus in a completely different, differentiated cell in the connective tissue, turning it into a pluripotent stem, calling it induced pluripotent cell. But so he got the Nobel, he was awarded with the Nobel in 2012. But practically, the result was useless because it stopped the cell cycle of the cells. So those cells are blocked in a multiplication phase. So if I use them, they keep multiplicating, and then we have to reprogram them to make them stop. So theoretically, of course, that was very important. But in our case, we got the same effect without genetic manipulation. So our cells remain cyclical, they don't change. If we stop giving them information, providing them with information, they keep multiplicating. So life actually is much more complex than any multiplication. as Yamanaka did, and as medicine is being doing now. It manipulates. So with tumors, gene therapies should be positive, but of course results won't be that significant because the system of reference in medicine is based on a wrong paradigm. Medicine is nature is much more complex. As far as anti-aging is concerned, our factors, besides keeping on the four genes that Yakinoma treated, they also keep on other genes that activate some certain proteins, preventing the senescence, that is to say, the cell aging. So on one side, we increase the cell life, blocking the telomerase cut. But anyhow, the cell would age anyhow. And we would just get old. But here, those factors, besides making life longer, they even stop aging. So life, you see that life is complex, and we just have to follow how our life acts. So here, we still have some time. So concerning mind and body. Those are the other factors that we studied in their 5 stages. As you saw, at the basis of life, there is information. This information keeps our balance and keeps us healthy. Our system of reference explains us how our body works, and it is the psychoneuroendocrine immunology, which is quite a complex system, but it's not enough. So this PNEI has to be integrated in the program. Our mind-body system, that is to say, as human beings, we are cognitive system reacting to any stimulus coming from the environment, and it's in balance as long as it's healthy. And those stimuli affecting our body can be sound, physical stimuli, cultural stimuli, environmental stimuli, and so on. Professoressa Di Modugno talked about Freud before, and how we turn wrong information, such as money, in a stimulus, and all those stimuli affect our psychoneuroendocrine immunology system. And what do they change? Well, they actually change the cells with bioelectric, metabolic, epigenetic modifications. We checked which affect the cells of this system, changing gene expression. So those cells of the PNEI system produce hormones, cytokines, and so on, changing the balance of somatic cells in the whole body throughout cytokines and hormone networks. But the system does not stop here. This system also works thanks to those factors functioning for the PNEI system, and all our fears and instincts that Jung talked about affect the whole body, not just the PNEI system. And those undergo metabolic modifications, which are epigenetic, of course, leading to a completely different response of our body cells, which produce mediators changing the gene expressions in the PNEI cells so that at the end the cycle is closed. And the balance is maintained thanks to a very precise and widespread piece of information with no distinction between mind and body. There's no real distinction, but we are just a whole answering in a complex and organic and united way to our stimuli. Thank you.